I'd like to, on behalf of the Cancer Care Nova Scotia and the Beatrice Hunter Cancer Research Institute, welcome you this afternoon. Thank you all for coming. I see a lot of familiar faces in the room, but for those of you that don't know me, my name is Carmen Giacomantonio. I'm the Chief Medical Director at Cancer Care Nova Scotia. I'd like to begin by welcoming Ms. Peggy Davison, the inaugural Board Chair of Cancer Care Nova Scotia and the individual for whom the award is named. It's the inspiration from Peggy and people like her that bring clear vision to the rest of us to see and understand what is important and to come together to do the right thing as we are doing today. Thank you, Peggy. Now, before I announce the winner, of whom most of you in the room know, kids kind of pretend a little bit, but so I'd like to provide you with some background of this very important and prestigious award. As everyone in this room will agree, research is one of the pillars on which excellence in patient care is built. Research provides us with new insights and information about cancer risk factors, thereby strengthening our efforts in the area of prevention. We've also taken steps, as you know, with the appointment of Dr. Louise Parker as the Atlantic Path Project Care and Canadian Cancer Society Research Chair. We at Cancer Care Nova Scotia and the CDHA Foundation were instrumental in creating this position, yet another important position in cancer research. Research is enabling us to make earlier diagnosis at a time when interventions can become more meaningful and more effective. Research brings new evidence-based treatments to the bedside and clinical arena. Research enables us to organize our systems to assure that people with cancer have better access to existing treatments and facilities in a timely fashion. Research is enabling us to wage a war, and a more effective war, on a disease, or really 200 or more diseases that we call cancer. And of critical importance, building a dynamic research program, as we are doing here today, greatly enhances our ability to attract and retain the brightest and best cancer specialists and scientists the world has to offer. I think, again, we have succeeded. For these reasons, Cancer Care Nova Scotia created the Peggy Davison Clinical Scientist Award in 2004 to support a clinician scientist in developing his or her area of research and to contribute to and improve cancer control in Nova Scotia. The Peggy Davison Award provides $100,000 per year for 3 years to be used as direct support for the program of the clinical researcher. It can be renewed a second 3-year time pending successful reapplication. The first recipient of this award was Dr. Eva Grunfeld. Eva is a professor of medicine and a national leader in health services research. Health services research attempts to bridge the gap between what is and what ought to be. As a result of the Peggy Davison Clinical Scientist Award, Dr. Grunfeld's research provides information to improve the delivery of care and access to cancer care in Nova Scotia and around the country. The Beatrice Hunter Cancer Research Institute administers the award for Cancer Care Nova Scotia. It managed the international review process that took place and began last fall. It's my great pleasure to announce Dr. Jason Berman as the successful recipient of the Peggy Davison Clinical Scientist Award. Congratulations to Jason. I want to take a second to congratulate and welcome as well Ben and Jake, Robin, Jason's wife, and Ruth, Jason's mom, who came from Toronto to be here and celebrate with Jason and the rest of us today. Dr. Berman is a pediatric hematologist-oncologist at the IWK Health Center. He's a scientist with a primary appointment in the Department of Pediatric Medicine and cross appointments in the Departments of Microbiology and Immunology and Pathology at Dalhousie University. Dr. Berman obtained his medical degree at the University of Toronto in 1997 and then completed his residency in pediatrics at the Hospital for Sick Children in Toronto from 1997 to 2001. Jason then moved to Boston for his clinical fellowship training in pediatric hematology-oncology at the Children's Hospital, Harvard Medical School. He completed his postdoctoral research training at a laboratory of Dr. A. Thomas Look in the Department of Pediatric Oncology at the Dana-Farber Cancer Institute in Boston. There, Jason gained expertise into zebrafish genetics and an appreciation of the power of using this novel model system to study blood and cancer development. In the summer of 2005, Dr. Berman brought his knowledge and experience to the IWK Health Centre and Dalhousie University in Halifax. Over the last 5 years, Dr. Berman has established an independent zebrafish research laboratory. His research is unique in Canada, and he is linked to an international network of researchers. All 5 of them get together every now and then. No, it's more popular than that. At this time, I'd like to ask Dr. Berman, Cancer Care Nova Scotia's Peggy Davison Clinician Scientist, to come forward and share some highlights of his research. Jason, welcome. And congratulations.
Thank you, Dr. Giacomo, for those kind words. For those of you that know me, talking is something that I enjoy doing, and the opportunity to have such a captive audience truly is a thrill. I'm really delighted to be here today and truly humbled by receipt of this award. I particularly want to thank Ms. Peggy Davison for her unwavering support of cancer research, and I'm really honored to become the second Peggy Davison Clinician Scientist. This award marks a significant milestone in my journey as a clinician scientist. When I arrived here from Boston almost 6 years ago, I had a vision of building a cancer research program, but was uncertain exactly how I would go about doing this. I knew I had excellent training, lots of enthusiasm, and some unique ideas, and the IWK and Dalhousie seemed to be the right place. There was opportunity for funding, tremendous support from colleagues and peers, and research infrastructure to help create the foundation of a state-of-the-art research program. This award recognizes our lab's accomplishments to date and provides the opportunities to launch our research into its next phase and translate our findings to improve the health of children like Olivia Mason, who you will hear from her and her family shortly, with cancer throughout Nova Scotia and Canada. I wanted to share with you a bird's eye view, or perhaps I should say a fish eye's view, of some of the highlights of the work we have been doing and how we'll be moving it forward. My lab uses the unique model of the zebrafish to study blood development in cancer. The zebrafish is a small tropical fish, about the size of a guppy, that originally hails from the Ganges River in India, but that you can purchase at your local pet store. Amazingly, these small fish share many of the same genetic makeup that humans do. They have all the same blood cells as people, and the same genes that control the processes of normal blood cell development. This is really important, as we now understand that leukemia and most other cancers result from abnormal development, creating an immature cell that divides uncontrollably. If we can better understand normal cell development, we can better understand how this goes awry and leads to cancer. We can then target these specific abnormalities within the cell, leading to improved treatments with less side effects. In addition to having many of the same genes and cells as humans, the zebrafish also produces large numbers of embryos that are fertilized externally and transparent, which allows us easy access to study developmental processes. We also have a number of tools and techniques that we can apply to the fish and then clearly visualize directly what's going on inside, as evidenced by some of the pictures you can see on the far wall there. We have taken advantage of these opportunities afforded by the zebrafish in our studies. We were the first laboratory to describe mast cells in the zebrafish and show that they function like human mast cells. The mast cell is an important immune cell involved in allergic reactions and inflammation, but more recently has been recognized for its role in promoting the spread of cancers. And it does this through its role in initiating an inflammatory response within the body. Our studies will allow us to use the zebrafish to visualize how mast cells contribute to cancer aggressiveness and test drugs on the fish that interfere with this process, leading to new drugs that can be used along with standard chemotherapy treatments to help patients. We have also been modeling human cancers in the zebrafish. We have been doing this in 2 ways. In some cases, we put the human cancer-causing genes into the zebrafish embryos. The genes get incorporated into the fish's genetic makeup and then will be passed on to many future generations of fish. These fish then develop cancers that look a lot like human cancers, such as acute myeloid leukemia, which accounts for 20% of childhood leukemia and is the most common leukemia in adults. Acute myeloid leukemia, or AML, is a disease that Olivia knows personally, and she will tell you about some pretty intensive treatment with some pretty nasty side effects that's required to treat that disease. With large numbers of these leukemia fish embryos, we can then screen vast numbers of chemicals in a fast and efficient way in order to find promising drug candidates that stop cancer development and kill cancer cells. Our goal is to find new treatments to help children like Olivia. In the fish, we can do this in a cost-effective and rapid manner that is challenging in more traditional animal models, such as mice and rats. And there are precedents for doing this. In other types of myeloid leukemia, such as chronic myelogenous leukemia, a disease that initially required patients to undergo a bone marrow transplant for control and for cure, can now be treated through Gleevec, which is a tablet that really targets a specific genetic abnormality in the cell. And so we're all searching for the next Gleevec, and I think the zebrafish will help us do that. More recently, we've actually been putting human cancer cells themselves into zebrafish embryos. We have done this with leukemia, but also with breast cancer and sarcomas, which are childhood bone and muscle tumors. We are able to see these human cells grow, divide, and spread in the zebrafish. In fact, we use a mutant zebrafish called Casper, like the friendly ghost, I'm dating myself with that reference, that is completely transparent without any stripes. And that allows us to help visualize these cancer cells, which we label with a fluorescent marker, and they can easily be followed as they travel throughout the fish. We have shown in some of the leukemias we have studied, where we know of an effective treatment, that by adding this treatment just to the fish water where those embryos are, that we can stop the cancer cells from dividing. This is very exciting, because if known treatments work in this system, it sets up the zebrafish not only to screen for new cancer drugs, as I described before, but also to be used to test a patient's cancer cells for responses to a particular treatment. Established drugs and new potential agents can all be used in the zebrafish to study their effects in real time. This is something that has not been easily done before. The zebrafish is really the ideal system for these kinds of studies, as we can directly observe what's going on inside, and importantly, we can see how the cancer cells are interacting with other cells in the fish, such as the mast cells I talked about, in the context of a living animal. This is something we can't do when we test cancer cells in a petri dish. As you can see, over the past 5.5 years, we have been developing some creative tools to study AML and other cancers in the zebrafish. We are now poised to use these fish models we have developed to identify new treatments, and the Peggy Davison Award provides the support to help move these efforts forward. We are now linking with pharmaceutical companies to screen new drugs in these fish model systems. The most promising drugs we identify can then be tested in other complementary models, such as the mouse, and then, through my involvement in clinical trials groups, such as the Children's Oncology Group, I can be directly involved in moving these discoveries into the clinic to help patients. Thus, this work is truly bench-side, or really tank-side, to bedside. The Peggy Davison Award also enables us to continue to recruit the best and the brightest from across Canada and beyond to work with us, fostering the training of the next generation of leading cancer researchers right here in Nova Scotia. At this time, our lab is the only one in the country using zebrafish in this way. Given the tremendous potential of the zebrafish model, I believe that the Peggy Davison Award will be instrumental in establishing our laboratory as a pioneer in these innovative approaches and a national leader in cancer research. Most importantly, I have every belief that our efforts will ultimately help find better targeted and less toxic treatments for children like Olivia and for children and patients with cancer across the province and across the country. I feel very fortunate today. I hope you will agree that my decision to come to Nova Scotia was the right one, and I want to thank Drs. Jonathan Cronick, Bob Bortolucci, Jerry Johnson, Dorothy Barnard, and Conrad Fernandez, some of whom are in the audience today, for recruiting me here. I have been welcomed here and provided with unparalleled opportunities, but it has truly taken a village to make this all work. Firstly, I have had the opportunity to work with a great bunch of trainees and researchers in my laboratory. They are energetic, dedicated, inspiring, and keep me on my toes. I have learned more from them than they have probably learned from me. They are the ones who do all the work, and as such, this award is as much for them as for me. Many of them are here today, and I want to thank them for coming. In particular, I want to mention Sahar Das, who has been my right-hand woman for the past 4 years. I have also had great mentors and collaborators here, and many of them are in the audience today, in Boston and elsewhere. They have been encouraging, supportive, and proud of my accomplishments, and in particular, my receipt of this award. This work would not be possible without the support and understanding of my clinical colleagues, both physicians and nurses, many of whom are here today, who enable me to balance my research efforts and my clinical responsibilities. I really would like to thank them. The Sobey Connors family was unable to be here today, but was very proud of my receipt of this award. I want to thank them for their unwavering support of my research. I am proud to honor their son Matthew's brave and fortunately successful battle with cancer through my research efforts. I also want to thank the folks in research services at the IWK as well at Dalhousie, who are always such strong cheerleaders. I feel very lucky to have them in my corner. Not to mention my administrative assistant, Jocelyn Jakes, without whom I would probably be buried under a mountain of paperwork. As important as support is at work, it doesn't mean much if one isn't supported at home. This is the emotional part. My wife, Robin, and my sons, Ben and Jake, are here today, and I know they are my biggest fans. Their patience, understanding, and unconditional love have provided the strength to allow me to pursue my research ambitions. Thank you. These ambitions and work ethic are instilled at an early age, and I have my parents to thank for their commitment to my education and sparking my scientific curiosity. I'm very grateful that my mom is here today from Toronto. I know she is so proud of me, as is my dad, who isn't here, but with me in spirit every day. Finally, I want to thank my patients and their families, and in particular Olivia and her parents, for their courage and fortitude. You teach us all so much every day and remind us of why we do what we do and why we need to make cancer treatments better. I want to thank Cancer Care Nova Scotia and the Beatrice Hunter Cancer Institute for this award, and particularly Peggy Davison, and the tremendous opportunities that this award will provide. Thank you very much all for coming today.
Jason is an amazing clinician scientist, and I feel proud to call him colleague. The things you see in what we're talking about today make him a great clinician scientist. The emotion you saw as he spoke make him a great physician. So, okay, thanks, Jason. Thanks for sharing all of that, your research, a glimpse into your world. I know the things you do will eventually translate into cures for people with acute myeloid leukemia, and your efforts will translate into better collaborative partnerships in Nova Scotia and around the world. I'd like to take a minute to introduce Dr. Jonathan Blay, the Scientific Director of the Beatrice Hunter Cancer Research Institute. Jonathan is going to pass a few words of wisdom and explain to us how this partnership is going to make us a better place.
Thank you, Carmen. I'm not so sure about wisdom, but I will be brief. I think we've heard indirectly today how important children are, and I don't want to allow too much of a delay before Ben and Jake are able to work on their own creativity through doing shadow puppets with a projector. It's really been a pleasure to work with Cancer Care Nova Scotia to bring the 2011 Peggy Davison Award to this decision. For those of you that are not familiar with us, the Beatrice Hunter Cancer Research Institute was formed in 2009 with the intent of working in collaboration with a strong network of partners in Nova Scotia and throughout the Atlantic region. This network includes fellow research institutes, charitable foundations, and agencies such as Cancer Care Nova Scotia that focus upon patient care. Here in Nova Scotia, we have a talented, internationally renowned community of cancer researchers, some of whom you see amongst you. And this is a resource that can and does benefit the province in terms of its healthcare decisions, attraction of key clinicians, and direct economic impact as a result of the research activity. The collaboration of different researchers leads to new ideas and stronger strategies, and within our institute over the past 2 years, we've already benefited immensely from the involvement of Dr. Berman as a researcher who can bridge between scientific experiments and clinical treatment. We look forward with great enthusiasm to working with Jason in the future as we combine our efforts to benefit both medical research and the mission of Cancer Care Nova Scotia in patient care. I congratulate Jason on receiving this award, both personally and on behalf of his fellow researchers and clinicians who are part of the Hunter Institute. He is here after having succeeded through a very rigorous competition that, as Carmen mentioned, involved review of his file by international experts and intensive interviews. The large audience we have here today is, of course, partly in recognition of the importance of this Clinician Scientist Award, but I know is also because of the respect with which Jason is held, and the importance seen in his work by those of us who can understand what he's doing and the unique advantages and potential gains that can be made by working within a tissue that just happens to reside in a trophical fish that's a member of the minnow family. So I'm very pleased to congratulate Dr. Jason Berman, a very worthy recipient of the 2011 Cancer Care Nova Scotia Peggy Davison Clinician Scientist Award.
At this time, this is a little impromptu. I'd like to give Sahar Das a chance to come up and present Jason with a little token of their appreciation and congratulations.
I want to say congratulations for Jason, and from all of our lab members, everyone. We have been honored to work in his lab, and it has been like a family for me, so congratulations.
As Jason said, Sahar is his right-hand woman and kept him on the rails this past 4 years, heading in the right direction. I've been holding this back all day. Jason, this is my little cliche, but Jason is now the big fish in his little pond. And I couldn't figure out time to say that. Now's as good a time as any. But seriously, I want to thank all of you for taking time out of your busy schedule to come here and attend this announcement. This is a very important announcement and a proud day for Cancer Care Nova Scotia, for Peggy Davison, who we're so thrilled to name this after, and for the province of Nova Scotia.